Hello, everyone. Today I'll be presenting on embedded multimodal sensing chips. This is a collaborative effort between University of Central Florida and World Precision Instruments. I will be going around the outline, that is our approach, motivation, whereas the microfabrication process, conclusion, acknowledgement, and future and acknowledgement. The pharmaceutical industry is in crisis because the most of the drugs before reaching to patients, it fails. 90% of the drugs compound fail when it reaches to the clinical trials. So, because the primary reason for this is because the models are based on animal testing and not the human relevant technologies. Recognizing this, FDA came up with an act into law where they mandate to get to end the animal testing, to end the animal testing and come with a better relevant technologies where the human responses can be studied to a particular drug. But we need better predictive models. For this, the best approach is having organ on a chip platform where you can have a microfluidic system where the lining of the cells with the cell type, a particular material with that particular cell type, the fabrication process, as well as the stimulations provided to the organ on a chip platform can give us electrical readouts. Those readouts can be in optical sensing, mechanical sensing, and electrical sensing. Uh, Is this a loose connection? So there are, when it comes to electrical testing, there are various modes of electrical testing which are done on an organ on a chip platform. And one of such is impedance cytometry, where a single cell passing through the electrode, we can, changing the alternating current, we can see what's the size of the cell or the membrane is. When it comes to amperometric oxygen sensing, the oxygen diffuses, resulting into a measure of the current, and we can measure the oxygen level. When it comes to the field effect transistor-based pH sensings, the hydrogen ions on the gate channel shifts the threshold, resulting into a current measurement. And when it comes to the transepithelial endothelial electrical resistance, that is TEER, it basically detects how the barriers are between the cells. Are they tight or loose? But one thing is lacking between all these. Most of the electrical platforms is embedded electrical readouts. We will be focusing on the TEER, where the TEER is basically, imagine a brick of a cell where where this mortar is holding up the cells, and it tells us whether the cells are tied together or they are leaky. And it combined with an electrical impedance spectroscopy, we can have, instead of passing a single frequency, we can sweep through a frequency and measure the resistance. These are various, when these two electrical measurements are combined together, what we can measure is barrier integrity, adhesion and spreading, evaluates the cell moment and tissue repair, as well as observes transient changes in the cell morphology. But this sensing field, this platforms has a sensing gap. Where, as you can see on the photonic sensor chip is placed on a away from the cellular source. We lack the spatial and temporal uh, data. And also in the optical readout, what happens is the cells absorb the photons and then we take those data, which is not that ideal when it comes to the data analysis. And when it comes to the sensor integrated PCB, there is scaling up is a challenge because there are big microfluidic systems which are outside the channel, having the oxygen and the pH sensing together. Uh, so our approach is to have a scalable multimodal platform where we can have an embedded readout. So this is basically fabricated electrodes, and we evaluated through three different materials, that is gold, platinum, and indium tin oxide. This is a transparent platform because indium tin oxide is conductive as well as transparent, which is good for the microfluidic systems or organ on a chip platform. These can have measurements of electrical readouts such as impedance. We can have transparency and the uh, cell observing cell over a period of time, having continuous and real time monitoring. This is the wafer level process which we did for each of the materials. The picture depicts the metal deposition of a gold, where we have these on a glass substrate which is transparent, and we spin coated the photoresist, have the maskless photolithography, pattern the design, and have the lift off done. The best thing about this, or the worst thing, as you can say, is having one design with for liftoff is okay, but having multiple designs is a challenge because you face short circuits, or the liftoff is messy, you lose all the devices in one go. So the optimization of the maskless photolithography, having the laser to put that particular dose combined with the intensity focus offset was very much important so that we can have a clean liftoff, which can have a stable stitching, have a clean undercut profiles, as you can see in this image. These are the electrodes which we fabricated from those are gold electrodes, platinum electrodes, and indium tin oxide electrodes. So this is the packaging we used because we wanted to test whether these electrodes after fabrication are testing on a lab setting, whether we are able to get the readouts or not. So we came up with an idea to have 3D printed rectangles filled with a PBS solution and put the devices under test to see whether we are getting impedance or not. Uh, so as you can see, we had the silver paste used on the contact pads to have the wire come out and have the impedance readout. This is the picture representation where it was very difficult because these are tiny electrodes, very tiny features. This is the packaged wafer. So the characterization we did, it was having impedance readout. As you can see at the low frequency, the area is barrier dominated. As you go on the higher frequency, it's media dominated. Basically, it's when you consider this model as it gives when we say barrier dominated is basically passing a frequency, low frequency, and seeing whether the cell are holding up tight or not. And when we move to a high frequency, we see how the cell membrane is. Sorry. We also had a TEER value measurement at 12.5 hertz because at lower frequency, we wanted to observe whether the cells are tight or not. So we calculated the TEER value with the effective exposed area by the 3D printed area of the electrode. We got these values, baseline values with the PBS, and we also had a transmission uh, transmission done for a different pellet of materials, and indium tin oxide stood out. So in conclusion, we have a wafer glass platform which is scalable, multimodal, as well as different materials for different studies. The future work includes having not only TEER, but also having oxygen sensing as well as pH sensing on one platform. So we have altogether an embedded electrical readout. And also having a biology test done. Thanks to my professor as well as the industry partners. And this project was funded by NSF. And we also got featured on UCF Today article for developing this electrodes, which further our partners could scale up in a semiconductor industry. Thank you. Okay.